Yeah, so now we are proceeding to the third question. Third question is,
yeah, but without
how
the question
do tumor
mark.
cells shape the immunomicroenvironment in human disease?
Thank you very much, and I wish to thank you, Ethan and Yonai, and of course, Isaac, having invited me here. I didn't bring pictures from the old times when I first met Isaac. I just want to make a personal note. As some of you may know, I was born without a father, and I don't want to say that the age difference would allow Isaac to be my father, but I was also born without an older brother. And Isaac certainly, in some respect, played this role and was very instrumental in the way I decided and I chose my research projects. 20 years ago, I was happily working on FC receptor, as some people here, Israel Pesht and Isaac may remember. And it was simple, there are only 4 FC gamma receptors. Isaac told me the important thing is tumor microenvironment. So that when Isaac said something, I had to follow it, and so I did so. And with some colleagues such as Eric Tartour, Jérôme Gallon, and Catherine, my wife, we decided to go into the microenvironment, and we found out that it was very complex. And since it was very complex, it was a bit too complex for our minds, and we decided that we should approach it by high-throughput analysis and computer analysis. So what I will do again, last sentence, then you can have the count of the time starting, is that I will do that as I was always done in meetings where Isaac invites me. I will present non-published work. And usually after I have a discussion with Isaac, and either I never present them again, or I work more to make them more interesting. As an introduction, what the last years have shown is that the tumor microenvironment is very complex. And you find all the inflammatory and immune cells, this is what interests us, that you wish it to look at. And what is seen, this slide is made for us to show that the immune microenvironment is maybe look complex, but it's not a mess. It is very organized. You have cells inside the tumor core, the tumor center. You have cells in the invasive margin. And you have lymphoid islets, which act as tertiary lymphoid structures, where we believe, and I have no time to discuss it today, the anti-tumor immune response is generated. So you have mast cells, DCs, you have T cells, B cells, whatever, name them. So we recently undertook with Jérôme Gallon a thorough analysis of all the cells that you can find, immune and inflammatory, in the immune microenvironment. And higher the peak is, more the density is high. So you see, in blue is in the center of the tumor, and yellow in the invasive margin. You see that macrophages are the most prominent cells that you can find. And it is followed by T cells mostly, B cells in the tertiary lymphoid structure. Some cells, the regulatory T cells, are not very present in terms of density. However, if you ask the question, what is the correlation between the clinical impact, if there is any, of the cell density and the cell density, you see this is well known, that macrophages, in red is a deleterious impact here on disease-free survival, have a deleterious impact. Having many macrophages is usually not good, but it's not so strong as compared to its density. FOXP3 cells, very rarely found, have in colorectal cancer a strong positive impact. The most important cells are CD3 cells, memory CD3 cells, memory CD8 T cells, and the B cells in the tertiary lymphoid structures, whom we know now do produce antibodies to tumor-specific antigens. So several years ago, we proposed this, what is called now the Immuno Score, in which we enumerate 2 markers here, CD8 memory cells, CD8 a row, in the invasive margin and in the center of the tumor, So when you are high for each marker in both regions, it is very good. These are early stage colorectal cancers treated by surgery alone. These patients have been cured. You see, we are 10, 20 years later. However, when you are zero, low, very bad, 80% of the patients have relapsed. These patients need other treatments. They need additional treatments. I may speak a little bit, maybe not so loud. But the most important part is the following. If you do a Cox multivariate analysis, the immune score is the only one that is statistically significant when the classical TNM classification is not anymore significant in terms of disease-free survival, overall survival, and disease-specific survival. And this, now everybody accepts this data, but I can tell you that this 10 or 15 years ago was not accepted as a, let's say, dogmatic, should be put into the thinking of oncologists. Now it has changed, and we recently published a meta-analysis of 20 different cancers, 124 papers, and as you see in most of the cases, there is a very good agreement that then when there is a strong memory CD8 T cell infiltrate, potentially cytotoxic, with a Th1 orientation, it's good. Blue is good clinical outcome. It is not so for the other T cell subsets. Treg, Th7, and Th2 are in half of the cases associated with bad prognosis, and the other half associated with no impact or good prognosis, and I believe, I will come back to it, that it depends on the overall inflammatory environment. So, well, there are a few exceptions. And also I learned that from Isaac, what is important is to be interested in the exceptions, among which there was Hodgkin lymphoma, but maybe the most striking exception was renal cell cancer. And we revisited the renal cell cancer. There were a few papers from Japanese groups essentially showing that CD8 T cells were bad prognosis in renal cell cancer. So we analyzed a series of primary and metastatic renal cell cancer, and we found that high density of CD8 T cells was correlated with low disease-free and overall survival, both in primary and metastatic renal cell cancer. So I will not go further in this so far. We have analyzed that and shown that it is due to PD-1 expression on the T cells, LAG3 expression, and PD-L1 expression, and PD-L2 on the tumor cells. But we ask the question, what is responsible for that? Is it the tumor or is it the soil? And to do that, we analyzed in the same organ, the lung, metastasis either from colorectal cancer or renal cell cancer. And I will very briefly summarize these data. We ask the question, does it recapitulate the primary tumor? And the answer is yes. In colorectal cancer, high-density CD8 T cells in the metastasis is associated with good prognosis. In renal cancer, cell cancer metastasis, it is associated with bad prognosis, and these are months from the date of the surgery of the metastasis. So from this part, what we conclude is that different malignancy feature different immune contextures, and that the phenotype of the tumor cell, rather than the hosting organ, is responsible for these differences. And we ask to ourselves, since now they are beginning of having definition of molecular cancer subgroups, not based on immunology, but based on genes overexpressed in different subgroups of the same cancer, do these also feature different immune infiltrates? And this is completely not only unpublished, it's the first time I speak about it. So to do that, we defined on 2,000 samples immune signatures. And I cannot see the colors from here, but we could identify signatures different from B cells, for T cells, for lymphocytes, for neutrophils, for macrophages, for endothelial cells, etc. And this is more details. You see that we have signatures for T cells, B cells, NK cells, B cells here, myeloid cell lineage, neutrophil, macrophages. And these signatures we first assess that they really correspond more or less, and this is reasonable to the density of the corresponding cells, and this is the density of mature Antigenicity cells. This is CD8 T cells, I think, and this must be macrophages. So there is a reasonable correlation between both. So if one analyzes the signature, one can have an idea of the expression of the genes and of the density of the cells in the tumor. So first of all, we use this signature. This is on the lymphocyte signature. In green are the genes that are correlated with good prognosis, and red with bad prognosis. And we ask the question, in available public databases, thousands of tumors, what is the impact of immune genes? In colorectal, 84% of the lymphocyte-associated genes are linked to good prognosis. In renal cells, 75% are linked to bad prognosis, and there is a even distribution of the other genes. So now we apply that to colorectal cancer, and there have been 7 different groups that have defined subgroups of molecular cancer, of colorectal cancer, based on genetic signatures, not of immune origin, and this is being merged now in a consensus signature. We got access to one of this, who separated 6 groups. An MSI group, MSI is microsatellite instability. These tumors have DNA repair mechanisms that are impaired. They produce a lot of peptides, and it is well known that these tumors are of good prognosis and have a strong T cell and CD8 infiltrate. Look at this group, stem cell-like group. This is cells that have undergone epithelial mesenchymal transition. I'll come back to it. And then you have KRAS and the others here. The group C4, here, stem cell-like, is the worst group in terms of disease-free and overall survival, whereas MSI is the best one. We apply our immune gene signature to that. What we found is that there are immune, so first to this cohort of 566 tumors, and then it was validated on a cohort of 900 tumors and most. You said that there is a strong expression of immune genes in 2 subgroups, C2 and C4, the best prognosis group and the worst prognosis group. C1 and C5 are quite dull in terms of immunity. C6 is like normal colon. But they are not the same. Myeloid cell genes are expressed in C4, whereas mostly cytotoxic genes are expressed in C2. And here you have a strong fibroblast and endothelial signature in C4. If we look more precisely into genes of these signatures, C2, the good prognostic gene, as genes expressed and involved in attracting chemokines that attract T cells, as well as interferon gamma, a Th1 gene signature, as well as checkpoint inhibitor signatures, PD1, PDL1, PDL2, and LAG3. And it is in this group that the scarce responders to anti-PD1 treatment are found. On the other hand, C4 has a strong expression of genes involved in inflammation, angiogenesis, and complement together with the lymphocyte gene. Why is it so? What shapes that? So we analyzed a series of tumors for amplification in red and or deletion of genes involved in chemokine production. Look here, CXCL13 is deleted. When CXCL13, which is produced by the tumor cell, is deleted, you have less B cells, you have less T effector follicular helper cells, and it has a bad prognosis. So when the tumor cells do not produce CXCL13, you don't have tertiary lymphoid structure, and it is a bad prognosis. Another example, IL-15. IL-15 has been shown deleted in some tumors. It is produced by the tumor cells again, and when it is not produced, it is a bad prognosis. So it is the tumor cell that decides, that produce the factors that are necessary to shape a good or bad immune response. I told you that group C4 had a fibroblastic signature. And so we analyzed, there was a correlation between the fibroblastic signature and the endothelial and the myeloid cell signature, and the answer is yes, not with the cytotoxic cell signature. But a myeloid-like signature is not proper to colorectal cancer. So we analyzed this signature in a series of different cells, breast, colon, lung, ovary, kidney, whatever, and we found it in cells in the various pan-cancer cells. Is it due to the cancer cell or is it due to microenvironment? So to do that, we analyzed a series of cancer cell lines from the cell line library. And on the left side, you have mesenchymal cancer cell lines, including 3 colorectal cancers, compared to the non-mesenchymal ones, which also in blue have the other colorectal cancers. And there is a series of genes that are overexpressed on these cell lines. And these genes, if you look at them, are the genes that encode for inflammatory, immune, and angiogenic molecules. Indeed, if we ask the questions, are these genes expressed in our various subgroups of colorectal cancer, the answer is yes, they are overexpressed in C4. And the reason for which C4 has a strong inflammatory invasive endothelial signature, it is because the tumor cells do produce the corresponding chemokines. One word on renal cell cancer, much shorter. The same trick has been done. Renal cell cancer has been classified by Jessica Zuckman-Rossi in 4 different subgroups. Group C4 is the group which has the worst prognosis in terms of disease-free and overall survival. It is also the group which has the strongest gene expression for B cells, cytotoxic cells, dendritic cells, T cells, and angiogenic cells. This is due to hypomethylation of genes in the tumor cells involved in regulation of T cell activation, of immune response, cascade apoptosis, caspase apoptosis, and chemotaxis, and it is associated to a strong infiltration of the corresponding cells that I just talked about. When we analyze the genes that are overexpressed in this group C4, I cannot read for here, but again you will see that you have the proper genes which are expressed in lymphocytes and myeloid cells, in inhibition and angiogenesis, etc. When we compare the cell line in tumoral tissue from kidney cancer, they're overexpressed versus non-tumoral tissue. And in the cell lines, you find them overexpressed, not in epithelial colorectal cancers, but also in these cancer cells that have undergone mesenchymal differentiation and have the stem cell-like phenotype. So it is a pan-cancer situation, and not only has the gene expressed in these cell lines, but also the proteins are produced. You see VGFs here, IL-6, IL-7, etc., IL-17, and so on and so forth. So the conclusion from what I have tried rapidly to show you is that there are 3 basically different types of cancer. You have the immunogenic type, the colorectal classical type, in which you have antigen, the MSI-like, you have everything you need to be immunogenic. This, the right IL-15 being produced, CXCL13, you have tertiary lymphoid structures, you have CD8 T cells, this take care of the tumor. Maybe later, or maybe very early, this tumor may escape, and these are the dull tumors. They don't express neoantigens. You don't have a T cell infiltrate, and these tumors are immune down. They are not of good prognosis. But you have the inflammatory type, in which you have antigens, you have all the goodies, but at the same time, you produce high angiogenic factors, IL-6, GM-CSF, TGF suppressor factors, and you have a disrupted immune microenvironment, and this also is of bad prognosis. And I think this is important because you will not address the same immunotherapeutic treatments to that. When you have everything you need here, you want to push it a little bit more by vaccine, anti-checkpoint antibodies. When you don't have T cells, you have to bring them. by engineered T-cell bispecific antibody. But when you have an inflammatory angiogenic context, which is dictated by the tumor cell, with VEGF production, IL-6, etc., you want to make the fire cease first by anti-suppressor, anti-angiogenic factors before you come with the other approaches. And I just want to summarize a few people, so many people who have participated. Romain Remarque, no, Etienne Becht has been the one who has mostly participated in that, the group of Jérôme Gallon, and of course Catherine Sotes Fridman was sitting here. And with that, I thank you for your attention.
Thank you very much.
Eva also.
Thank you very much for a very interesting talk. Thank you for a very interesting talk. One point is not clear to me. What is your definition of immunogenicity or antigenicity?
So, we have what is the definition of immunogenicity? So the definition of immunogenicity should be the proof that there is naturally an immune response going on against tumor-associated antigens. So we could show that in terms of antibodies, when we took out the B cells from when there are tertiary lymphoid structures in non-small lung cancer, where we could show that indeed they produce antibodies to tumor-associated antigens. For T cells, the repertoire is very large. We couldn't show that. So my operational way of defining is a T cell, Th1, CD8, memory T cell infiltrate which positively impacts clinical outcome of the patient.
Eva.
I would, now I take the opportunity to give my lecture. So I have many remarks, but one is a criticism. Pull yourself together. You didn't mention Cd4 cells. And this is coming out of my work, and that's my lecture. I mean, we are concentrating as the immune response always to make the cells to be killed. So it's the CD8 cells, and we are our cells in our laboratories a little bit responsible for that. Recently, I was interested in seeing that the Cd4 cells are very important because they make a lot of lymphokines. Now, and also we look at the agents which are the causative agents of the cancer, but we don't look so much about the differentiation of the target cells. How does it respond to this stimulus? Because some combinations from the point of view of malignancy don't work. So what Leo Saxon, this is my lecture actually, because it starts, of course, the lecture starts with the microenvironment, but the microenvironment also influences differentiation. So Leo Sachs made the notion of differentiation therapy in myeloid leukemia cells in mice, and saying that if you change the differentiation of the cells, you will inhibit malignancy. So could imagine that with all these chemokines and lymphokines, but forget about the T cells, they don't need to kill the cell. They just change the differentiation. I mean, you have so many versatile tumors, I wouldn't say that. Certainly it works for some tumors. Then you change the differentiation, and then it doesn't care for the stimulus. It doesn't grow. So please think about that. Anyway, in your next lecture, please take Cd4 cells also. Okay.
Questions to Eva's lecture? Good.
And the Cd4 cells have a major role to play. They also are very diverse. And you see among the Cd4 cells, you have the Th17, you have the Tregs, you have the Th1, the TFH. And what we begin to really see is that the impact will be different also, and that's what you said, depending on the rest of the microenvironment. a regulatory T cell can have a very positive impact on clinical outcome of a patient if it takes care of chronic inflammation which is going on around there. So I agree, they play a major role there.